yesterday, we had 2 lectures. We had a keynote lecture for Samuel Igea, and he was delving on computational discovery of antimalarials. And he was showing us a couple of mutations that he had planned to investigate for dihydrofolate reductase of Plasmodium falciparum. And as explained, it was an ongoing project, and there was a lot of excitement and questions about this project. And later on plan, we had today's first talk that was supposed to be Dr. Marco Yukic, who was supposed to speak this morning at 9 o'clock. So he rather gave his talk yesterday and presented CMDoc, which was a tool that they had developed in their lab to carry out molecular docking. He clarified a number of questions and the ambiguities that have been raised regarding molecular docking in general, and also presented us a tool. And in the hands-on sessions, we had 2 hands-on sessions. We had 2 hands-on sessions. We had HS01, which is Dr. Yannick Fenang Jumbo was presenting a tool that he had built to predict agrochemicals. And those who were very interested in bioinformatics, even though this time around his talk was more towards chemoinformatics, while Dr. Yannis Konst was presenting also open protein binding site Robix, which is a tool that he has built. And those who were participating in these hands-on sessions were able to run parallel. And that's going to be the same today. We are sorry that yesterday, at the beginning of the day, some had difficulties with the link because of some technical problems, which was fixed. And now we are pretty sure that today is going to be a smooth day. So as you know, the first oral presentation today was supposed to have been done yesterday. So I am very excited to introduce a new speaker, who is Dr. Martin Medrad, and he is going to talk in the place of the first presentation, and then the second presentation will continue as it's supposed to be. So Martin, you are going to ready to start, and you are supposed to speak for 50 minutes maximum, and we are going to have questions and answers for 5 minutes. And then after that, we're going to continue the rest of the day. We also have another speaker at 9:40 that's not available, and we are going to replace him. I'm going to let you know who's going to replace him, because we had some late speakers that submitted abstracts that were put on waiting list. So we are going to choose and announce to you before that time who is going to take the place of the speaker that's supposed to speak at 9:40. So Martin Medrad, you have the floor. Please share your slides and you can start, please. Yeah, we can see your slides, but apparently you are muted. Are you not muted? Just unmute yourself, and then we can hear. We cannot hear you. Hello. Yes. Hello. Yeah, we can hear you now, please. You can start from the beginning. We were not hearing you from the beginning. Yeah.
Okay. Hello, everyone. Good morning. My name is Martin Medrad. I'm very grateful to participate today in this training workshop explaining on computational approaches in secondary metabolites. My research was molecular dynamics on the structural stability and supervision of Rebecca in organic solvents. The supervisors was Dr. Stephen S, Lucas Paul from Medicine, and Professor Crouch from Miami University, USA. Based on my research title, the background of the study that this in the systemic drug that includes a group of medications called topoisomerase 1 inhibitors, and this drug is used to treat several types of cancer, including colorectal and ovarian cancer. Despite its effectiveness, this is associated with several challenges, such as limited solubility, poor bioavailability, and toxicities. Recent studies have to prove the pharmacological properties, but greater attention has been given to its stability and observation behaviors. Therefore, understanding these properties is crucial for unlocking full potential in drug design and discovery. Here you can see the structure, the Ntie-Kang, and this has got 33 carbons, 38 hydrogens, 4 nitrogen, and 6 oxygen. Therefore, you can see here the structure, the 3D structure of this drug. The main objective was to investigate the influence of solvents on these physicochemical properties of Ntie-Kang range of solvent by computational methods. But specifically, the objective was to analyze the effect of polar and less polar solvents on chemical formations and the structural behaviors. Secondly, the specific objective was to evaluate solvent energies of Ntie-Kang selectively polar, less polar solvents. What the methodology approach I used observing this the influence of solvent on this the drug structural behavior. The first procedure was to prepare the topology, in which I said the phosphate, which I used the tip4P water model, but also I used I defined the atom, bond angles, and the dihydrogens by using the phosphate. Here was the general hamburger phosphate, and also I defined the molecular type and the bond of the atoms, referring to the structure that I showed previously. Secondly, I defined the box and surveyed the molecule, which I defined the box size, which were in 4.6 nanometers at 298.15 Kelvin, but also I filled with water, which was modeled by tip4P 2005 model. But also, the third procedure, I did energy minimization, in which this procedure was done in order to assemble the solvated neutron system in order to remove all the caches that were made during simulation. The third one was to equilibrate. The system was equilibrated with itself and it brings the temperature simulation. But thirdly, I did production MD, which I collected the data for simulation. And the last procedure was analyze the trajectory, which I used the trajectory visualizer software, Travis, in order to see the data that I collected for simulation. So the first results from failed calculation. This one was for Van der Waals contribution that I looked for the effect of this Van der Waals contribution to the free energies of Ntie-Kang in the range of solvent. Therefore, high DMSO, acetone, methanol, ethyl ether, water, acetonitrile, and DMF. But you can see here, water showed the highest peak. This was due to the high intramolecular forces in the water molecule. Therefore, solvent intramolecular interaction within the water molecule were higher compared to intermolecular interaction. Therefore, the water molecule tried to pull up the cavity in order to break down those bacteria within itself. That's why the van der Waals contribution for water molecules seem to be higher. But also we can see for cyclohexane was more negative, in that there were high intermolecular interaction that contributed to the salvation advantages of Ntie-Kang cyclohexane. But also when we come to the phase calculation for due to the Coulombic contribution, you can see here the water molecules had the most negative phases. This was also due to the presence of the strong hydrogen bonding within the water molecule. This led to the most negative Coulombic contribution to the salvation of this Ntie-Kang, the water molecule. But in addition, this field question were done by using the multi-state Bennett acetylation method, in which we applied all the data. Those were in the unit of 80. But also we did the total phase calculation results, in which we compared the van der Waals, the Coulombic contribution. Therefore, you can see here, and the lowest one was for pyridine, but also DMSO, also for DCN. So you can see that those are the solvents which had the less hydrogen bonding interaction to have the lower total energy contribution. But after that, we did the structural analysis results in order to see some contribution due to the structure of this solvent interacting with the drug. Therefore, you can see that we did structural analysis by using CMAT, which this aimed to gain an overview of the inter-solute solvent interaction, in which here you can see the results for acetone, which were among of the solvent, in which we have the two parts, in which the left part shows the interaction connection between the promising proton donor and the acceptor, while the right hand side of the heat map displays the measure of the interaction strength. Therefore, you can see here, this left color shows that there are higher interaction strength due to hydrogen bonding. So we can see that the atom of the drug H2 and the oxygen 1 are the strongest molecular interactions. But also when it comes to cyclohexane, you can see here we have the blue color. So from the heat map, which we can see that the blue color is at the highest distance and the highest distance, but also the shortest distance. Therefore, this proves that there is some moderate interaction. Therefore, from this one, we can prove that those atoms which had the strongest interaction had the purple color, which proved that there was strong, there was a contribution of hydrogen bonding the interaction with the drug molecule. Therefore, from that, from this structure analysis, we can conclude that the higher relative solubility by cyclohexane, but also there was slightly insoluble in the water at the room temperature. But also cyclohexane proved to be the best solvent for solution of Ntie-Kang. But also thirdly, we can conclude that the solution of nitromethane was relatively low due to the absence of this hydrogen bonds. But also hydrogen bonding has a strong influence on the solution process. But also the last we can conclude that the drug solubility and the structural stability depend on the solvent used. Thank you very much. That's the end of my presentation.
Okay, thank you very much, Martin. That was quite a good talk. You have been able to investigate the presence of the effects of different solvents on a drug. And the question we can ask is, before I open the floor, what are the potential applications of this study? Do you see the potential applications?
The potential application of this study that we can use, the pharmaceutical industries can use the data from this study to manufacture the drugs by using the specific or the best solvent. The best solvent which can produce the drugs with a high efficacy.
I see. So, but has this drug not yet been experimentally, I mean, the solubility of this drug in the ferrous solvents has not been experimentally done already? What was the goal of doing a theoretical study?
And this drugs have been experimental, but also comes for the issue of the efficacy and the best solvent for manufacturing these drugs. Therefore, it comes to the issue of the solvent, which is more appropriate for the production of these drugs. Therefore, from my study, I can see that the best solvent depends on the issue of hydrogen bonding which are present from the solvent and during the sorption process.
So is this drug, I mean, this drug is already in the market, right?
Yes.
Is it administered as a tablet or as a suppository or as a syrup? How is it administered? Is it by injection? How is the drug administered? Let me ask.
And this drugs can be administered orally, but also.
So okay, it's administered orally. And your interest, yes. Your interest is at the level of absorption at the intestine, or where exactly is your interest?
My interest was at the level of the absorption, but during the preparations.
Okay, during the preparation, because okay, the drug is administered not as a tablet; it's prepared before administration.
Yes.
But certainly, you would not want to administer this drug together with some organic solvents, right, to the patient. I mean, I don't see you administering this drug to a patient in TMSO or some other organic solvent, right?
Yeah, exactly. Because this solvent was this solvent was for preparation at the pharmaceutical industries.
Okay, that's
not for
during the
when you
synthesis or
the patients.
Okay, this is not for administration.
During the synthesis.
Yeah, okay, because it was not very clear at the beginning, so I was questioning myself. Anyway, the floor is open for questions. Please just raise your hand, and I will call you to ask a question. If you are not able to talk, you can write your question on the text, and we are going to ask. So we have quite enough time for questions. Can you raise your hand if you have questions on this topic? If so, yes, Daniel, please.
Yes, it's not a question, but just an additional audio asking just to supplement. We know the solvent moderates the molecule in different ways, and solvent play a really important role in the conformation of stability of the molecule, and also issue about the polymorphism about the molecule. So ideally, the work was trying first to figure out at the molecular level using it as a model molecule to establish and trying to understand how different solvents they could moderate natural compounds or even synthetic compounds. So this was first to get this and getting a microscopic view of how the solvent would moderate and bring the atomistic behavior of it. Then this we can translate to several other.
Yeah, for sure. the compound drug we have right in front of us has several functional groups capable of making hydrogen bonds. And if you are going to model it with solvent that has no propensity for hydrogen bond formation, it's certainly not going to be possible, right? You certainly don't want to model it something like hexane or something like this.
There was a communication breakdown, so can you repeat it again?
I was saying that the molecule I have right in front of us has several functional groups basically of forming hydrogen bonds. And the second thing is there is possibility of forming intramolecular hydrogen bonds and intermolecular hydrogen bonds. So, which means that in essence, you have to narrow down, you certainly want to narrow down the number of the kind of solvents you're going to use. You probably want to use only polar protic solvents. So, what solvent categories were used in this study, or you're just at the beginning?
We used a link from polar to non-polar to polar. So we did a different solvent, different polarities.
Okay.
And we wanted just to get an insight of it, because we know an example.
Okay. So was hexane, for example, among your solvents?
Yeah, it was there.
Yeah, because from every indication, the parts of the molecule that would likely interact with hexane are only parts that are hydrophobic, right? Because you don't expect hexane to interact with all these hydrogen bond forming groups because of the fact that hexane will not form hydrogen bonds.
Yes, sure.
Yeah. There is a question here. During your extraction process, how many solvents did you use? Question is because I don't think you guys extracted the molecule, right?
No, this is not exactly. We
that's what I was thinking that the question was framed. Yeah, are there any other questions, please? If there are no other questions, I am very excited to introduce the next speaker to you. Next speaker is Dr. Gemma Turon. Gemma is the CEO of Ersilia Open Science Initiative based in Cambridge and in Barcelona. And she's also co-founder of this organization. And this organization has been very instrumental in developing countries to make AI models accessible to not only generate AI models, but also train scientists on how to apply artificial intelligence for drug discovery process. Recently, we have been collaborating for like 3 years already, and they are also collaborating with many other institutions in Africa, including H3D. So, Gemma, you're very welcome to give your talk, please.
Thank you, Fidele. Can you see my slides?
Sure, I see your slide very well.
Perfect. Okay, hello everyone. First of all, thanks for the invitation to present here, especially invitation from Fidele, with whom we've been collaborating for the last 3 years. I am Gemma. I'm a doctor in molecular biology, transition later to computational biology, and I'm currently the CEO and co-founder of Ersilia, where we focus on developing open source models for antimicrobial drug discovery. So Ersilia, as I said, is a nonprofit initiative. We focus on global health research and aim to provide AI tools to infectious disease researchers, mostly in Africa. Why we do that as a nonprofit mission? Later on, I will share more on the science, but basically, as you know, this is the world map. And when we look at the communicable disease burden, unfortunately, as we all know, this affects much more low and middle income countries, particularly those in sub-Saharan Africa. But on the contrary, when we look at the research that happens worldwide, we know that most of the research funding and research publications are coming from the global north, which has led to a huge imbalance in what is being researched, because 6 of the top 10 causes of death in low-income countries are still infections, but most of the research is focused today in non-communicable diseases, and we are really lacking new treatments for diseases like malaria, tuberculosis, but also neglected tropical diseases like schistosomiasis and cryptosporiasis, and so on. So what can we do as a small nonprofit initiative to revert this? Well, we use artificial intelligence. Artificial intelligence is cost-effective compared to experimental research. It's fast to implement. It's data-driven, meaning that we can reuse data that has already been produced and it's out there. We can also reuse methods that have been developed maybe in the context of non-communicable diseases like cancer or Alzheimer's and apply it to the context of infectious diseases for which there is usually less data and it's less research. And finally, of course, it's a growing field. We are scientists and we are really excited to be working at the state-of-the-art, with the state-of-the-art new technologies. So now going into the science, we classify our toolbox of AI models in 3 categories. The first thing is we serve AI models that predict bioactivity, that predict toxicity, that predict ADMET properties of compounds. That is what we call the Ersilia model hub. So we make these models easy to run and use for researchers. The second thing we do is we build some of the AI models come from the open source domain. Others are built by ourselves with this tool that I'm gonna share more about in a few slides that is called Shadrack. And finally, we are now really looking into generative AI technologies to expand the chemical space in search of new entities with good activities against the pathogens. So let's start by the Ersilia model hub. Basically, what we aim to do is to take care of all the steps that go inside training an AI model, which means we get data, we process it, we train the model with this data, we test that the model is actually working, we validate it, if it's possible external with collaboration with experimental scientists, and then we add this model that we call the deployment layer, where basically a researcher can select a model of their interest, for example, against a specific pathogen, input a drug and get the output. In this case, in this example, would be the target of this drug. It could also be the predicted IC50 of killing a pathogen and so on and so forth, right? So we aim to take care of all this process that happens inside so that the scientists can really focus on their research and use these tools as an auxiliary and complementary to their experimental pipelines. As I said, we have 3 kinds of model in the hub. The first one are models that are already out there, have been developed by others, but are basically sitting in a publication and not really being used, or even the repositories where these models live are difficult to actually follow and implement. So we take these models. In this example, I'm taking this very nice paper on antibiotic drug discovery, where they developed a whole new AI network to basically predict bioactivity. So we take this model, we incorporate it in our hub so that it remains there and is easy to use. And basically you can select it, input a drug, and it will tell you whether it's an active or an inactive according to this cell publication. We also look for relevant data sets in the literature or in databases such as Campbell or PubChem. In this case, I am showcasing an example with a large open source screening of a library of compounds for malaria. And we use this data to train a new model and basically also provide this model to the community so they can simply input a drug and get whether it will be an active or inactive antimalarial. And more importantly, we develop AI in collaboration. So with our partners' data, I'm here putting an example with the University of UEA, which are interested in natural product activity. So we try to build models that work on the natural product space and that are able to predict activity, for example, against a specific pathogen or virus. So these are the 3 kinds of models that we can find in our hub, which currently has around 150 models. This is how it looks today. And it's constantly evolving and improving. So we are very happy to hear feedback or people wanting to use it. It's all open source and free to use for everyone. And I just want to showcase where can this be useful. I could not put this beautiful picture. Some of you are here in person. I'm very jealous of that. That's the Center for Drug Discovery at the University of UEA, Cameroon, with Mount Cameroon at the back. Whereas you all know Professor Fidele is focused on using African natural products as a source for novel antivirals. And in this case, we can use our hub of models to screen these libraries of natural product compounds, and then hopefully select the best leads for testing. And this is coupled not only with computational analysis, also more traditional techniques like docking, and finally experimental testing that is also supported by the Wistar Institute. So this is the first part of what we do, is provide these models and train scientists on how to use them. The second thing that I wanted to highlight today is this pipeline to build AI models that we have called Zierep. We have developed this in the context of a collaboration with the H3D Center in the University of Cape Town, South Africa. So what does it involve at very general level to build an AI model? Some of you maybe attended yesterday's hands-on session, so probably know about this. So basically, we take a library of compounds that are here, with their associated activities, so active, inactive, against our assay of interest, let's say killing the E. coli bacteria. These molecules, of course, cannot be read by the computer because they are actually images, 2D representations. So the first thing that we do is we featurize, meaning we convert this nice structure that we can visually as chemists understand into a vector of numbers that for us is not that interpretable, but much easier to read in the computer. So this first step is called featurization. And once we have these features, we simply we train a neural network or random forest or other machine learning architectures to basically associate these vectors with their activities, so that when a new molecule comes in, we also featurize and convert it to these numbers, and then the model will predict whether it's an active or inactive based on what it has learned. At a very, very concept level, you can see that this vector, this simplified vector, of course, is similar to the vector of this molecule. So probably this model will interpret that this molecule is also an active, right? So that is what is happening behind the scenes. It's not that more complex than this. So there are two parts that we really need to take into account when we want to model chemistry data sets. The first part is the featurization, and the second part is the fitting of the model. So in our pipeline that is Zierep, what we have created is an automated way of selecting an ensemble of chemical descriptors. So not just only one, but several of them. These are plugged in from the Ersilia model hub, so they can be easily interchanged. So from when we have a molecule, of course, the molecules get standardized, and then we select the chemical descriptors of our interest through the Ersilia model hub. We get all these vectors and basically pull all of them together, input where there are missing values, normalize when they have to be normalized, and we get a very comprehensive description of this molecule for the computer. Once we have done this, we go on to the second part, which is the actual training of the model. We do that with a series of automated machine learning techniques. Why automated? Because we want these tools to be maintainable, to be easy to update by people that may not have all the knowledge on artificial intelligence yet, that they are still learning, so they can just use these automated tools that will decide the best model architectures. So we use a combination of tree-based classifiers, of neural networks. I'm not going to go into detail today because for the sake of time. We also use some dimensionality reduction techniques to extract the most important features and use them with classical machine learning techniques. And then we try to also start introducing new things like image-based representation of the molecules. So we have all these AutoML systems, and with that we create a final model, which is basically an ensemble of all of these. So how we have applied that at the H3D Center? This is University of Cape Town, another beautiful mountain, Table Mountain. So the H3D Center, for those of you who don't know it, is a drug discovery center that focuses on anti-malaria and anti-tuberculosis drug research. They have a whole array of proprietary data that they have been collecting over the years. And basically, we provided them with this tool to model and create a virtual screening cascade that mimics what they are doing in the laboratory. So they can reproduce their steps computationally and hopefully save time and money in selecting which molecules to progress. This piece of work was just published last year in Nature Communications. So if you want to know a bit more in detail how we build this CAiSMD pipeline, you can go ahead and read the publication. It's open. So basically, this virtual screening cascade at H3D has assays for whole cell assays, data for cytotoxicity, and for ADMET screenings. So we have used all their data and also sometimes complemented it with external data when there was not enough internal data. I'm not gonna go over all the figures, but just the main results. So what CAiSMD outputs is this kind of results canvas that I know can look very complex. I just want you to focus on one image, which is this one. This is the final score that you get. Each dot is a molecule. This is a test set for which we actually know the real activity of this molecule in killing the tuberculosis, the Mycobacterium tuberculosis parasite. So here we have molecules that are inactive and how they have been ranked by the model. Here we have molecules that are active and how they have been ranked by our model. So we can see that really inactive molecules receive very low values, whereas active molecules receive much higher values, making it very easy for researchers to discriminate between which molecules may be good for experimental testing and which molecules maybe we could discard and not lose time in testing them and synthesizing and testing them. I also want to show you this figure because we've done this, as I said, at the scale across several assays. These are all the assays that we have modeled. So activity against Plasmodium falciparum and Mycobacterium tuberculosis, cytotoxicity in human cells, and then solubility, clearance, permeability, and inhibition of cytochromes, and so on. And these are the values, so the areas under the curve, which is our measure of how good the model is. For most of the models, we have very, very good performances. And you can see here indeed how these drop curves are actually looking very nice. Any value above 0.7 is actually quite nice, and we are most of the times achieving 0.9, 0.85 area under the curve values. And the last thing that I want to show you today, regards to CAiSMD, is how we can use this prospectively. So here we took experiment that they did. They had a library of 65 compounds, which they synthesized all of them and tested all of them against Plasmodium falciparum, cytotoxicity, and so on. So basically what we did is we tried to reproduce this with our modeling exercise, and we were able very easily to pick which ones of these 65 molecules were actually performing better. So this is the original molecule that started the chemical series. And here, just by eye, we selected a few of the molecules. For example, 37, where the predicted activity against our target, which is Plasmodium, was high. It was a soluble compound. It was permeable, and indeed, when we validated that experimentally, we see that it is actually a good candidate. For example, in this case, we have a good candidate because it has high activity, but the model predicted that it would be toxic. And when the X3 tested it, indeed, it was toxic. So this is how you can actually select molecules that have a nice profile according to your needs. This can be customized to your particular set of experiments, but basically it allows you to pre-filter libraries of compounds and save time and money in doing the experiments. So that's our second tool, which is building AI models and applying them to actual ongoing experiments. And the third is ChemSampler, which I am just going to give you a very, very, very short overview. This is still work in progress. This is where we are focused now. So this is the unpublished results and still pretty much playing with this. So basically, now instead of wanting to predict the activity of a new compound, what we have is the contrary. We have the activity and we want to predict the compound, right? We want a compound that has the activity that we desire. So we've started piloting this in the context of malaria and are moving now into HIV. We did that first for the open source malaria consortium, and basically we generated virtually generated drug candidates that had that were predicted to be active against Plasmodium. These are all candidates that the computer generated. They all come from the same chemical series because that was the restriction from open source malaria. And basically we selected 8 of them, for which 4 actually have very good in vitro activity, which proves that these AI generative models are really capable of generating, creating, identifying new molecules with good activities. So that said, and I'm just, I think, right on time. We are a non-profit initiative. We develop AI tools for drug discovery. We combine remote work and on-site implementation. We've been invited already twice to the University of WEA and can't wait to go back again. We also do capacity building activities and trainings. Everything we do is open source, meaning that it's free to use. It doesn't have any patent restrictions, just citation, of course. And we offer support to research institutions across the Global South. We're always happy to hear about other people's research and look for collaborators. So that's it. Of course, cannot finish without a thank you slide of all our funders and the people that has helped us get this far, as well as our collaborators and our amazing team. So I think that's it. I will stop presenting maybe. Yeah, thank you all.
All right, thank you very much, Gemma, for that nice presentation. We have just 2 minutes for any questions that can come up. Any questions, please? If you have a question, just raise your hand or just text on the chat.
I do have a question. Can you hear me?
Yes, Paul, please go ahead.
You mentioned AutoML. Is that actually that AutoML which is developed in Freiburg?
No, sorry, AutoML refers to a general framework. We use, for example, Flamel, which is from Microsoft, and AutoGluon, which is from Amazon originally. So we use a combination. AutoML refers to anything that is automated.
Okay, because there is also like a package called AutoML.
Yeah, I know.
Okay, yeah. And this is developed around the corner. I was interested if that is actually the case. All right, thanks a lot.
Thanks, Paul.
A question?
All right. Any other questions, please?
I have a question here.
Yes, go ahead, please.
Hello, yeah. I think thanks for the very impressive presentation. Quite insightful and obviously on the research. But according to the models you provided, actually using the conventional or drug discovery approach of the West. But now that you're working with the African, I mean, in Africa, you know that having been in Africa, you know that we know or from experience that African medicine doesn't use this one active ingredient. You'll find that there are several active ingredients that contribute to the efficacy of a given drug. How are you gonna? I mean, how do you intend to tackle such a complexity?
Yes, thanks for the question. That is a very relevant question indeed.
Can somebody just pause I hear sound coming from behind, so we can just mute themselves and allow only the speakers to speak, please. Can you guys all mute yourselves? Thank you. Thank you.
Thanks, Fidele. This question is very relevant. Indeed, the limitation of machine learning and AI for drug discovery at this moment is that you do need at least the chemical structure of the compounds, which is not the case for many natural product extracts, right? For which we don't have all the compounds that are inside. When we have the compounds, there is some ways that you can model interactions between those, and this is something that we are looking into. I don't have any example that I can show now, but this is something we take into account. Where we struggle most is when the natural product extracts are not fractionated and isolated into pure compounds. If we have the different compounds in an extract, then yes, we can model interactions between those. But when we only have the extract, that our tools basically are not useful in that context. So that's a limitation that we have to face.
And also, I mean, I don't think it is a limitation, but I think that's what you just said you're working on. I mean, you have activity of, the desire to have activity, and then you work towards finding your system or your model works towards finding the structures. Is that not the case?
So for the models to work, you need the structure because you need to convert these structures. With generative AI, you predict the structures, but then they may not be related to the natural products. Something that we are really trying to do, focusing on generative AI and thinking of natural products, which we know is of huge interest for research in Africa, is we are trying to focus on generative AI that provides us with easy to synthesize compounds that mimic those natural products that are usually very difficult to achieve, to synthesize in the lab or to extract. So this is the direction that we are going now, but certainly happy to talk more about all this offline, because I know we are over time and I don't want to take time off the next speakers. But thank you for your insightful questions. I really appreciate. And yeah, we are working on that and thinking how we can make our tools better for our users.
Yeah, thank you very much, Gemma. Actually, the next speaker is not available. He's traveling right now. The next speaker is Joel Onoja. He's a postdoc who is working with us from the first of batch, natural product chemist. And he's flying right away from Nigeria to Cameroon. Unfortunately, he cannot make it. So if there are one or two more questions from Gemma's talk, we can take it. And then Daniel and I are going to fill the gap to give you a perspective of what we plan to do in the hands-on session this afternoon. Please, if you have one or two questions more to Gemma, we can take it if Gemma is available. Yes. So,
yeah, just let me. Okay.
Yes, Jonathan, please. Yeah, Jonathan, go ahead, please.
Yeah, thank you very much for the presentation. Unfortunately, I joined a bit late because of the time differences. Most of the natural products which are active against parasites, it has been observed that some of these compounds, they are effective. Sorry.
Yeah, go ahead.
Most of these compounds are effective. Sorry. Against several targets of the parasite. That means the molecules which are effective against a parasite actually are effective against different targets inside the same parasite. So do we have a platform where if we have a compound, we can look at its effect on several targets in that parasite at the same time?
Yeah. Thanks for this question, Jonathan. The short answer is it is possible. The long answer, of course, is even more complex. So in a model, you predict whatever you have used to train the model. So if you've trained the model to predict with data from an IC50 assay, like a wholesale assay, you predict the activity of the compound to kill or to inhibit the growth, for example, of Plasmodium falciparum, regardless of the target in principle that you're using, right? Because it's predicting whole cell activity. That's what the model has learned. Then if you train the model with, for example, a data set that is focused on inhibition of, I don't know, certain kinases, for example, the model will provide you with the answer of whether this compound is inhibiting this particular target or not, or is interacting with this particular cytochrome or not. So if you want to know, if you want to identify the specific targets, you should use several models or a multi-model that predict the activity against each of the targets individually. This is possible. The problem is that for many of the infectious disease targets that we are interested in, there is not that much data. Unfortunately, unlike, for example, in cancer, where you have a ton of assays that will that you can use a ton of data sets around a specific target, inhibition of a target, or activation of a protein, and so on. So the short answer, yes, it's possible. The long answer is because there is very few data, except for some targets of malaria and tuberculosis. The others become really challenging to predict. I hope this answers.
No, thank you very much.
Really, can I?
Yes, you can, please.
All right. So just addition to Gemma was saying. So the question of Jonathan is what exactly we're going to do to the tutorial. So we will explain. We will do it not from a machine learning perspective, using like kind of a deep learning code to a single ligand with multiple protein. And we will use also a physics-based approach like molecular logging, to where you have a single ligand with several proteins from the same organisms. So we will learn it today. Thanks.
Yeah, so this is going to be hands-on session 04. So
Wow,
those
that's
who
great.
are willing to join can be very willing to do it. But we are not doing any injustice against hands-on session 03, which is also a very nice hands-on session with Paul Zierep. So it's just that the two will take place at the same time, fortunately. And the good thing is that we can give the option to record one of them and then we keep the other one live. Okay. Any other question?
Just a quick one.
Yes, please.
Yeah, Gemma, can you please reconcile your work with the drug repurposing?
Yes, of course. So basically, our models would be like an ideal exercise for drug repurposing in the sense that they allow for the quickest screening of approved compounds or compounds that are under development for other disease indications, and provide you with the leads for which other disease indications these compounds might be interesting. So this is just a matter of choosing the right models that predict, I don't know, activity against malaria, tuberculosis, and so on, and running large screenings. It doesn't, so what takes a lot of computational power is to build the models. It doesn't take much computational power to just run predictions. So you can select the models that are relevant, that you're interested in, and then screen compounds and see if any of those are providing signal for another target or another pathogen or another disease indication, and then start from there for drug repurposing. So indeed, drug repurposing is a perfect exercise for AI-based drug discovery.
Cool. All right. Thank you very much. I think it was quite interactive. We are going to pass on to, thank you very much, Gemma, for your time. And we are going to pass over to our next shared 10 minutes between myself and Daniel. Maybe I talk 5 minutes and Daniel can talk 5 minutes about what we plan to do today, so that those who are willing to attend the session will have an idea of what it's going to be like. And so I will give you basically what I plan to do. I would say, yeah. I am sitting see. Okay. So can you see my window now? My slides?
Yes, Fidele.
All right. So we are going to talk about searching the natural product database. Specifically, we are going to be searching the African natural product database in this exercise, which we have put together over the past 10 years. And so I would do like 20 to 30 minutes of hands-on session where we are going to be practicing on our computers on searching the molecules that are in this database, the medicinal plants that are in there. So basically, I will talk about the compound identifiers. We'll talk also about searching by keywords and similarity search, and as well as searching by compound name. So the various search options we have there in this database. And I will describe how a compound can look like, what are the information available about every compound database, what are the information available for every species there, and how we can connect this database to other databases, how you can search, for example, moving from the ANPDB to another database and so on and so forth. And our exercise is going to be on a subject we call chemotaxonomy, which means the classification of medicinal plants or plants in general according to their chemical contents. So we are going to do a quick exercise on this. And of course, those who are going to be working here, you can use any browser, and with any browser, you can actually get access to the ANPDB on this, using this link. And with any browser, you can also get access to the tutorials. We have tutorials available online, also information about what you can download, how you can contribute to the ANPDB, and so on and so forth. And so will show you, for example, how to search, for example, with compound identifier, if you would be willing to use the ANPDB for your search, is because I know most people who are citing the ANPDB are using it for feature screening to identify bioactive compounds that can potentially bind to specific targets, and they go ahead to isolate these compounds and test them in the assay. But you can also do a quick literature search using this platform. So it's very friendly, user-friendly platform. For example, if you have a situation like compound name here, you can simply see that if you want to know how many compounds contain, for example, the quercetin core, then you realize that quickly, a quick search there will tell you that about 134 compounds in this database contain this quercetin, for example. And then you would now be able to see that for each of these compounds listed here, it would tell you, for example, the PubChem ID, so which means that if you are set to search this compound in PubChem, you can simply click on the blue link there. And then it can also tell you how many species are there. Are there any species in North Africa or East Africa? For example, we are building now, so the data for the other regions are not on the platform, but we already have data for North Africa and East Africa on the platform, and so on and so forth. You can also, with this same search, for example, if you're interested in cancer, you can just by typing cancer, you will get compounds that are potentially related to any form of cancer, which might be compounds that have been tested to inhibit some the growth of cancer cell lines, or it can simply mean compounds that have been identified from medicinal plants that have been traditionally used to treat cancer, or some form of inflammation or something like this. So information like this available. If, for example, you are interested in virus, you can type HIV, for example, it gives you general information on keyword search. You can type, for example, any other disease, perhaps malaria, so you can still get the same information. If, for example, you are interested in molecules that have a certain degree of similarity with an compound of interest, by simply drawing a structure here, for example, this aspirin, by simply drawing a structure like this, you can then do a substructure search. That means the algorithm will simply help you to find compounds that contain that same structure, or a similarity search. That means compounds that have similar properties with this one in terms of motor similarity. The idea would be that, typically compounds that are similar will bind to similar targets and things like this. So in essence, there's a lot that you can do with this database. So if you are really interested in attending this session this afternoon at hands-on session 4. We are going to be doing practice of this, and I'm very glad to share with you whatever questions you may ask. I'm going to be there with Thomas Musioka, who is also going to present the South African Natural Compound Database and with some vitro screening results he has had before. And I'm also going to be there with Daniel Madulu Shadrack, who is going to be talking in a brief moment. Thank you very much for your time.
Thanks, Fidele. Not presenting anything, but I think I will present during the hands-on session. So after surveying how we can obtain those compounds, and then we will learn how to do molecular docking using two approaches. Using a deep learning approach with a model that it does not need already trained, but wherever you want to go, it does not need to have a pre-training set again. That is based on compression neural network. And then we will use another approach based, for example, on VINA. So I will demonstrate two approaches, and then we will see how we can do target fishing in drug design. I'll try to highlight on how we can move from either hit identification to read optimization, but in just the highlight. This is what we are going to do in the end session 4, and we are going to answer Jonathan's question that was asking. Okay, so those who are interested in that kind of thing, you are welcome. And yeah, Derek, do you want to say something?
Yes, I think we are pretty cool on this because in the next talk, Paul Zierep is going to be giving an insight of what he plans to give in the hands-on session 03. So the two hands-on sessions that we have this afternoon, and it's from the University, the Institute of Informatics in the University of Freiburg. And he and I have just crossed our ways one time because I was spending 3 months in the Stefan Gunter's lab in 2015 as a Humboldt Fellow. And that time Paul was not there. And then when later on he came to the group, I was not also there. And of course, in 2019, I was in Freiburg for 2 weeks also. So he worked for some time with Stefan Gunter in pharmaceutical bioinformatics before moving to the Institute of Informatics, and he's been building and working on the Galaxy. So, Paul, you're very welcome to give your talk, please.
Let me see if I manage to share easily. Should be this one, right? And then slideshow. All right. Can you see my slide?
Sure, yeah. Yes, we can see.
All right, perfect. Yeah, thank you very much, Fidele. So as Fidele already said, I used to work in Stefan Gunter's lab. Did some things in my PhD towards genome mining and all that. It's been a few years since that I moved around a little bit, but now I'm working at the Galaxy team in Freiburg, which is a rather big team that is using Galaxy for many different things and also maintaining the biggest server in Europe and the second biggest server overall in the world. And I am in the metagenomics department, or I'm basically the lead of the metagenomics department here. But I think there are some very good and nice features from Galaxy that can also be used for secondary metabolite discovery. So I thought it's a great opportunity to give a short talk here and also hands-on session, because Galaxy is always rather used than looked at. So I will go a little bit about the motivation why I want to talk here. Try to give a short primer in Galaxy. It can be very complex. So everybody who's really interested should join the handout session later on, where we can look in detail on some features. Then, I would like to present a little bit the community I'm running here and how I think that could merge or also support secondary metabolite discovery in some regard. I quickly built together a short workflow using Galaxy that I think could be very useful in this sense, and I will explain a little bit the idea behind this, and that will also be the basis for the hands-on session later on, and then I also give a short outlook. So I am working on a or like I'm leading or I'm co-leading a huge community called the Microgalaxy community that is basically targeted towards everybody who's doing microbial research using Galaxy. And we did a survey last year about our users on who's using Galaxy. And as you can see here, there's only a very small amount of people from Africa. It's not very surprisingly, since we're the European Galaxy server, but it's open to everybody around the world. And in my opinion, or in opinion of a few people here, Galaxy could be extremely useful or beneficial, especially to people from the global south, that includes Africa, for many reasons, which I will talk through in the next stages. So this is a bit of the main motivation why I want to give this talk here, because I think there is an audience that could actually really use Galaxy and have a benefit from it. So what is Galaxy? Before I say you should use something or it's something useful, you should maybe first know what it actually is. So Galaxy is a web platform that can be used to do bioinformatics, but basically to do any kind of data science by podcasting command line tools to a web front end, and also allowing to combine those tools into workflows. So it's also a workflow manager. And it is developed for more than 20 years, which is an incredibly long time for a bioinformatic tool and the overall workflow manager. And it's still maintained, and there's an increasing use base, and many people actually use it a lot. And there are much more use cases than just having like a simple interface to tools. And I will show them some later. So, but the basic idea is that you have a panel where you can choose tools. Every tool has a rep. You can use those tools online, and you can also process the data in something that's called a history, which is basically like a folder where your data is stored. And like I said, I can tell you many things about this, but it is much easier to actually use it and play around with it. And this is what we're going to do later in the afternoon. So if you want to learn something about this in detail, please join that session. So like I said, you can run tools individually and do your research as you do it with the command line, but you can also combine those tools into workflows. That's a very old figure. The workflow manager by now looks much better, but you can actually have a visual view of your tools, connect them together, create workflows from this, publish them, share them with other people, redo them. And I mean, everybody who's doing bioinformatics knows that one of the major parts that people do is building workflows based on tools. And what is extremely nice from Galaxy is that the user doesn't really have to care about how those tools are executed, especially in terms of resource management. So you use the front end, and then Galaxy knows how to handle dependencies for your tools. It also knows how to assign these resources. And I mean, that's a very complex task if you consider that there are tools you have a huge amount of resource requirements. So the European Galaxy server, so Galaxy itself is an open source project. Everybody can spin up their own server everywhere around the world, and there are hundreds of them. We have actually lost track of who's used the Galaxy. I mean, they're using companies, but also by a single person, institute, or whatever. But we are running in Freiburg the European server, and the European server has huge resource amounts, and those resources are freely available for everyone around the world. And for Germans or for Europeans, usually getting a cluster in university is not such a big issue if you want to run large resources. But I can assume that for many countries in Africa or the global south, this is much more difficult to get access to those resources. If you use the European Galaxy server, you get all of this for free. And yeah, I mean, all you need to do is log in, and you can run experiments that are not possible to compute at all on your own machine. So there's somebody talking in the background. Mirelli, could you please? Yeah, thanks. And we also joined forces in Europe to even increase those resource opportunities. So now the jobs can process all around Europe using the so-called Pulsar network. And this is, like I said, free for everyone to use. So definitely that's something that you should look into. So I myself, like I said, I'm working in the microbiome department now, so we moved a little bit away from secondary metabolic discovery. What we want to do, kind of support everybody who's doing any kind of microbial research, not only microbiome research, in Galaxy. So what we are doing here mainly is that we are integrating new tools. We kind of prioritize it because we have a lot of tools, and you only need to select the tools that are used. We integrated complete workflows, so high quality workflows, and we also create tutorials, so something that people can follow to learn and also conduct the research and use this as a basis for actual real research. And Galaxy is used for like actually a lot of things. I think our publication list of applications that use Galaxy is over 20,000 by now. So there actually there are a lot of use cases for that. And we are focusing on this regard for the microbial research domain. And there are some reasons why Galaxy is particularly useful for microbial research. First of all, I mean, there are some tools that have huge computation demands. I mean, you might not know many of them, but for example, Kraken 2 can easily use more than 100 GB of RAM, and I don't have 100 GB of RAM on my computer. Processing this in Galaxy is no problem. Parallelization, I mean, usually you don't analyze one sample, you analyze hundreds of samples, and that then can be easily conducted in Galaxy as well. Storage requirements, I mean, many tools require databases that can be hundreds of gigabytes or even terabytes. Difficult to do at home as well. And in any bioinformatics setting, there's usually a frequent update of tools, databases, which is difficult to follow for single groups or single person maintained workflows if you have dozens or maybe hundreds of tools. Galaxy has something that's called a semi-automatic update infrastructure, meaning that many tools and databases are automatically updated on Galaxy when the tool itself is updated. I could talk about it for a long time, which I will not do it, but you can benefit from it for free when using Galaxy. And the idea I had is why not bridge? So what you can do with Galaxy, you can bridge different domains because we have tool stacks of many different domains all in one framework, and you could just connect those if you learn how to. And I thought, why not bridge something like omics domains with something interesting like chemoinformatics, for which we both have huge tool stacks, but we don't really have combined workflows. And so I thought that would be a great opportunity to do secondary metabolite discovery, because it's basically genome annotation if you think about gene clusters, and chemoinformatics if you think about characterizing the predicted products. And so this will be also, I will show a use case how I made this in the afternoon. So really just playing around, clicking together things, and like with a bit fine tuning. And of course, I have some deeper knowledge of Galaxy. I think that could be something that could already lead to publications rather soonishly and be used large scale for some discovery efforts. So one thing I want to be using, therefore, is the chemical toolbox, which is something that I have nothing to do with. It's a former Ph.D. of us develops to the main part. So that's like just a summary word for a lot of tools that are related to chemoinformatics that were integrated into Galaxy. And you can use them for free. They're just there to take. And this is, of course, something you can use to characterize molecules. And I heard some keywords in the last talks, chemoinformatics, docking, and all this. And we have a lot of tools in this regard. I don't know them a lot because, like I said, I usually do metagenomics. But those can be used. They can be connected. They can be updated. It's basically all available. So that's what I thought. Let's lose it rather than redoing something here. And well, there are a lot of publications about this actually toolbox, but the main idea is to include chemoinformatics tools into Galaxy, and then Galaxy takes care of the resource allocation and the resources and all this, like I said, as well for this. So my idea was, maybe some of you were here already in my talk, I think 4 years ago, which was just before Corona at the end of my PhD. So at this time I worked with CAiSMD, which is a web server that does secondary metabolite prediction and then also characterization in a way, or screening. And I don't work on this anymore at all, but I thought, why not just recreate this in Galaxy to show if that can be done with Galaxy. And that is exactly what I will show you in the hands-on session today. So the idea is to use genome annotation or gene cluster prediction, which is done with Anti-Smash, which I think is one of the best tools for this, and probably the best tool you can use freely and integrate. And Anti-Smash is available in Galaxy as a tool, so you can just use it. And then we screen those products in compound databases to see if the compounds are similar to something we know, and that might be then an interesting bioactive compound. And I mean, that could be, for the long run, is that there's a database called Magnify, which is usually used for metagenomics data, but metagenomics data can also be metagenomic assembled genomes. So actually a source for gene clusters and their compounds. And I tried to kind of bring together what I did in my last PhD together with my work now here. And some of my ideas are that we could use this database, which is also some kind of integration into Galaxy, so we can kind of pull data from this database and get it into Galaxy and do some investigations in terms of secondary metabyte prediction or characterization together with metagenomics analysis. And one of my bachelor students, he recently integrated the Magnify pipeline to Galaxy as well. And we could use this actually to kind of investigate how metagenomic properties are correlated to secondary metabolite production and such. And I worked with a workflow that does machine learning based on metagenomic properties from this database. And I just thought, well, we could also reuse the same workflow, but instead of doing metagenomic information correlation thingies, we could also extract gene clusters. Like I said, we could use Anti-Smash in a workflow setting, predict compounds and do large-scale characterization of those and finding in compound databases. That's a bit of the idea of the workflow that I tried to set up. And that I can already describe in my Outlook. So the idea is a little bit to use genome mining based on metagenomic data, which would be really nice because it combines both of my works. And then we can correlate those data with different metagenomic properties like alpha diversity. So see how is the community dynamics infected by compounds, and maybe we can also use that to backtrack interesting compounds, because I mean there's always a kind of logic behind how those things work. You can also correlate strains, and we can also include metadata and many other things. And then, of course, there could also be different screening methods and such. So in Galaxy, you can basically put all this together as Legos and quite easily make up huge workflows that are actually very informative. Some other things we're also working on is to include maybe some dedicated tools. There's BigScape and BigMap, which is a bit of a gene cluster anti-smashy tool stack tailored for metagenomics, which we also want to include in Galaxy as tools at the moment. So one of those tools is already integrated to the main part from a bachelor student, just needs to be merged into the main repositories. So with this, I would like to thank you for your attention. And if you have interest in what we're doing here, please have a look at our project page, where you can find all the contact information. I usually put all the links to get in contact with me or our team in the slides, so when the slides are shared, people can find the necessary information. We have a huge training network, which is where you can find vast amount of training materials to get into Galaxy or any particular topic. And for the hands-on session later on, so the only requirement which we should try to do before we start a hands-on session would be that you register to use Galaxy EU. Be sure it's use Galaxy EU, because there are many other servers. Org, for example, runs in the US, and then you will be on the wrong server, so please make sure that you're on this one. And then, like I said, in the afternoon, I will guide you through how you can create a workflow in Galaxy for secondary metabolite discovery, and also a bit of idea how this could be extended. And you will probably see that when you follow the tutorial, that this can be used for any kind of research by now. And this is also something we're trying to do on a large scale effort in the Galaxy community, to go away from saying, okay, for bioinformatics, I mean, any tool, any research tool can actually be integrated in Galaxy and be used. And we're doing this, we're also going to material science and things which are completely of my understanding, but basically anything can be integrated here. But like I said, I saw that many people here talked about machine learning. We have a huge machine learning tool stack. We have chemoinformatics and a vast amount of tools which can be used, including free resource allocation and a lot of power, which like I said, nobody can really do at home. And if you don't have easy access to a cluster, that's definitely something to look into. And yeah, time to discuss maybe some aspects in a bit more detail. One thing, last thing to say, maybe I registered a TIAS, which is training infrastructure as training, which means that there are some resources from Galaxy allocated only for this training. So really try to join in, because that's actually something that is great to do. All right, thank you very much.
Thank you very much, Paul, for that exciting presentation. And we are very hopeful that this hands-on session is going to have a lot of attendance. In the meantime, if you have any questions, we have 2 minutes to take the last questions before we go on coffee break. Yes, please. Any questions? We have 2 minutes to take questions.
Maybe that will be more in the afternoon.
Yeah, so if it's that case, I will declare the coffee break open, and we will return. The coffee break is for 15 minutes. We will return at 10:35, and I am going to be co-chairing the afternoon session, which is going to be have a roundtable discussion on all the morning speakers and all the various ideas from yesterday. And so have a good time on break, and let's meet each other at 10:35. See you. Everyone, I hope you had a wonderful break. So the next point on our agenda, sorry for the delay, is going to be this roundtable discussion. We should bring together all these previous speakers, both those from yesterday and today. So the goal of this workshop is to, this roundtable conversation is to see how can we harness all the information we have gathered so far to real-time projects. How do you apply all the knowledge you have and the tools that have been made available for your ongoing projects and so on. So if you have questions regarding the speakers that have come today, or if you have suggestions on the way forward for a specific project, it's the time. Please just raise your hand and you are free to talk.
Professor Fidele, sorry, I wasn't available yesterday. Can you just give a quick recap of yesterday's?
I can hear you very well, please.
I'm saying, are you able to give a quick recap of yesterday's proceedings? Because I wasn't available.
Sorry. Yesterday, actually, we have everything recorded on YouTube, and we have everything recorded, so you can certainly follow later. But I would say in brief that there were two talks yesterday. We had a keynote speaker who spoke for 45 minutes. That was Samuel Egier, and he was basically showing some of the results they have had for discovery of secondary metabolites against Plasmodium falciparum dihydrofolate reductase, which is a very interesting target. And he was looking at how to simulate mutations on this protein, and which are likely responsible for resistant malaria. And he was showing exactly the number of molecules that have explained already, molecular docking, molecular dynamic, classical approaches, and of course, and simulating also the effect of mutation on these molecules that they identified, or the drug target interactions, or basically what he did. And the project is ongoing, so in essence, the results are still being improved. And he said that it's a call for us to return to natural products for malaria discovery. And of course, a lot of questions came. One of the questions which was very intriguing is how do you model simulation? How do you model synergistic effects? For example, if you are taking a plant extract which contains several molecules, and you want to say that, okay, the person is feeling better and all of that, what really is happening? Is it that all the molecules are interacting with the same target? Certainly, it's multiple target effects, and how do you model all of that? Of course, it was clear from the interventions, including that for Daniel, that with target fishing, it's possible to take a single molecule and in an array of proteins, see reverse docking, or what is typically called target fishing. In an array of docking of proteins, see which of these the compounds binding to. But then how about a mixture? Because typically plant extract, which used traditionally, like what aqueous extracts are typically containing several metabolites. So how do you model all of that? That was a question that we were dealing with yesterday on Samuel's talk. And after that, Marco was supposed to talk this morning, but he was not chance, so he spoke yesterday, and he gave highlights on what are the various things you should take into consideration when you're doing the docking. Was trying to more or less give it some guidelines on some pitfalls that can be the result from molecular docking if it's not properly guided. And he says there has been an observation that in the past years, so there are quite a number of errors in the papers that are published from docking. And he said a couple of things, including the unit for docking, docking scores actually, and binding affinities, docking scores and not binding affinities, and mentioned a number of things that you must take into consideration when preparing your protein. You must know the biochemistry, you must know the assay, you must know the tautomeric state, you must know the of the ligands you are talking, you must prepare the flexibility, take the flexibility of the ligand and put into consideration and all that. So he presented to us a tool he developed called CNDoc, which is available for free, which you can actually try your hands with. Unfortunately, he's not able to answer all the practical questions. But of course, there were 2 hands-on sessions after yesterday's keynote, and the hands-on sessions where one of them was on Probis, that's protein binding site 2, and the other one was Yannick, who was showing us the chemical, not chemical checker. Yannick was showing hold on, this tool is different tool he works with for predicting agrochemical interaction with targets and so on and so forth. So I was not a part of both of them. I just once in a while go to Yannick's session, but I was more on Bobby's hands-on session. So maybe someone who was part of the hands-on session 1, Yannick can throw some more highlights. Yannick is there himself, maybe. Yeah. Are you satisfied?
Yes, I'm very satisfied. And thanks for bringing me up to speed on that. I think I have a few comments on what you talked about and what Gemma and the other guy who talked about Galaxy has to say. Basically, they're saying we guys in Africa, we need to use these tools that are out there. That's something that really came on top of, caught my eye or my attention. But also, I also need to mention that, there are opportunities on this continent. And I also must mention that we are taking advantage of these opportunities. One good example is the South African, is it H3P or something? The center, the University of Cape Town, who are really using state-of-the-art drug discovery approaches. And the other group that is notable is that of Fidele, who are using computational and not only computational approaches, but also other practical approaches to discover potential drugs for diseases that affect Africa. Somebody else who's notable is Shadrack Daniel, the Tanzanian professor, who is also really applying computational approaches to solve real-time problems. So these are just things to indicate that there are opportunities and that researchers are taking advantage of this opportunity. But what you raised is that how can we apply all this knowledge how can we apply this knowledge to benefit current projects or running projects? But what I would also like to raise is that how can we apply or harness, to use your word, how can we harness this knowledge to foster collaborations and to start new and hopefully cutting edge projects? Thank you.
All right, thank you very much. I think I will get back to you very soon. I am sorry I did not officially introduce my co-chair. I already announced in the morning session that I'm co-chairing with Daniel. So Daniel, really, you want to turn on your video, and Daniel Madulu Shadrack is based in 2 institutions in Tanzania. He's also created a company, Phytos. Phytos is the company he co-created with his wife. And Daniel is a very dynamic person. I have known him for many years. Actually, we met for the first time in Italy in ICTP, the International Center for Theoretical Physics. And those 2 chemists who are found in the physics environment. And so we were talking a lot, and he invited me to Tanzania to come and participate in this training workshop in Arusha, in Nelson Mandela African Institution for Theoretical Physics, for African Institution for Science and Technology. Good. So I was there in Kilimanjaro, and we spent a good time together. And so thanks to Daniel, I was able to meet Lucas Paul, who is my first PhD student in Tanzania, who is now graduated, and he's now training students. I was so excited to see Medrad today. He's one of the trainees of Lucas. So more or less, I'm already having grandchildren in Tanzania. Now, Daniel have been working a lot in this area of training. So his training is physical, so he gets some funding to bring students from Africa to Tanzania, while mine is more virtual, and now it's becoming physical. And last year, Daniel was in Boya with his boss, also Hulda Swai, and they were together. They gave talks, and Hulda came a session in our workshop in Boya, where and Gemma was there as well. So Daniel, you're very welcome to co-chair with me this talk. So I'll get back to Stephen. Stephen, maybe you want to talk briefly about yourself, your institution, and what you guys do in general, and how we can see how these tools can be of help to your project. Stephen, back to you, please.
Yeah, that's me. That guy. So let me just put up the video. So my name is Stephen Barasa. I come from the University of Erick in Kenya. I'm a lecturer there. I've been there since 2015. But prior to that, I was at Rothamsted Research Institute in the UK. Prior to that, I was a WHO fellow in WHO TDR Pfizer fellow at the postdoctoral fellow in at Pfizer in the UK for a year to train in drug discovery. Prior to that, I did a PhD at the University of Manchester, organic chemistry, biological chemistry. Prior to that, I did an M.Sc in synthesizing mosquito repellents at Kenyatta University, but I was allied to the International Center of Insect Ecology and Ecology, CPE, Nairobi. Prior to that, I did at Kenyatta University. But basically, here in Kenya, the story is the same, but that narrative has to change. We don't have facilities for practical chemistry or for practical drug discovery, and that's why I took this foray into computational or using computational tools, because these are practical, time-saving, and quite relevant tools that we can mostly things, just open source things, and that's how I came to know people like yourself, Fidele, using these databases. We've got the South African databases, the Central African database, the East African. I mean, that's how I got to know all this, and also got into contact with Shadrack, the professor from Tanzania, who is very good at using computational. So in brief, that's my background, but we try and do a little synthesis, chemical synthesis that we're able to do, because I'm trained in that. And I also try to do medicinal chemistry, obviously because I'm trained in that. But obviously medicinal chemistry is purely synthetic chemistry, but we cannot do that yet. But hopefully using collaborations, be able to do that. I've had several. I've graduated one PhD student and around 3-4 master students, and I've got numerous amounts of, I mean, around 12, over 12, and over 12 students doing their fourth year projects at the University of Medrad. And I'm happy to for any contribution. Thanks.
All right, Stephen, thank you very much for the input. So I will be next week, March, I'll be in Nairobi. So how far is it from Nairobi?
It's not 5, just 6 hours. But
just 6 hours. It's half a day.
Yeah. 6 hours by road. 6 hours by road, but maybe 40 minutes by plane.
Okay, all right. Anyway, yeah. I was last year. I was in Nairobi. Of course, I'm almost like a citizen of Kenya. Almost. We have this African Drug Discovery Accelerator project, and this is where I find you interesting, and I've been invited to this meeting again next week, so I'll be there in Nairobi from the 4th to the 8th. Of course, the meeting ends on the 7th, and so I would see how it can be like. Maybe if it's a virtual meeting, we can do. I was also at Nairobi University of Applied Science, so where I am already doing some mentoring. Holiness Nose, Dr. Holiness Nose is also getting interested in this work. So last year I gave a talk there, and I was really excited by the questions. So we can actually do a couple of things together. You know, we're already working with Daniel, so we can do a couple of things together. Of course, you can always come to Cameroon if you want.
Yeah, okay, brilliant.
So that'll be great. So we have in the Center for Drug Discovery, there is already synthesis going on. There is natural product work going on. There is computational chemistry. We are now setting up biological screening. So we're now setting up biological screening. We have done the first screening, and more or less we think that we are moving forward. So it will be a good thing for us to harness our knowledge together.
Yeah.
Good.
Yeah.
Any other questions, please? Any questions specifically addressed? Yes, Daniel, please.
Thanks. It's not a question, but just want to add on what you was talking with our great mission to biophysics in Africa. I think I'll be having a great trial for a couple of months to come. Kenya, Uganda, can make it that as a one mission. And I think at some point, Gemma will be at some point.
Okay. So certainly we can have this. We always have a possibility of meeting. So just let me know the schedules, and then we will see how we can harness all that together.
For sure.
Yeah. So I would ask a question to Gemma. Can you talk to us briefly how these tools are being used, the various Ersilia Hub tools? I know that you already have working sessions with some of the students who participated in the training you did in Boya. So maybe you can talk to us briefly about the other institutions in Africa where you are developing. I hope I'm not trying to be skewed by looking at Africa, but I think there are also other participants from India, from other developing countries, which have found this workshop very, very exciting. So maybe you talk about the various institutions and how useful some of these are. I know you just published a Nature Communication paper with your collaboration with H3D. So maybe briefly how practically this is happening already on the continent.
Sure. Thanks for the question, Fidele. So what we have been doing, so Ersilia is 3.5 years old. So we dedicated these first 3 years to setting up the technology and, of course, getting it published, just to get peer review validation and everything. So what we are looking at now is we've identified, I think after these 3 years of being around and talking about with researchers and so on, the best way to start using our tools is to first join one of our training sessions that we've done in person in South Africa and in Cameroon, but many more are coming up for 2024. We are doing one in Zambia and hopefully more in West Africa. To do that, we partner with people like Fidele, also, for example, with the H3D Foundation, which is a spin-off of the H3D Center, just dedicated to training and capacity building. And after these trainings, like we did at the University of Boya, what we are doing is trying to provide follow-up to the scientists that attended the training. So just for you all to keep in mind, we offer training for people who is actively involved in research. This means master thesis, PhD thesis, postdoctoral research, and so on, right? Because we believe that only by actually applying the skills that you learn to an active research project, you'll be able to leverage them. So we do follow-ups on this project. So we've been working with the people that attended our trainings at the University of Cape Town, at H3D, at the University of Pretoria. We have a project upcoming at Stellenbosch University here in Cameroon with the University of Boya, but also with hopefully the University of Yaoundé first. So the idea is that we provide first an initial core training on our tools, and then we are able to do a follow-up that is either a full research project or simply more of a data science support, as we are doing with Professor Fidele's students after they attended the course. So we have exciting news because we are trying to put together a more comprehensive program to be able to allow more scientists to kind of join these trainings and benefit from this up to 1 year support after the training. So yeah, this will be the big news, hopefully for this year, but we're still setting up the program for that. So I can't share much more at this moment. But yeah, so institutions that we are trying to work with, also always spending on grant funding, of course, with Daniel, he has amazing training opportunities. So we hopefully are part of that this coming year. We are talking with, of course, Kemri, for example, in Kenya, and so on. So I'm very happy to hear about other people with whom we might want to collaborate.
All right. So that is really exciting. So what would be the conditions for the team to welcome you guys?
To welcome us. So no, for training, we prefer to do trainings that so in-person trainings, 1 to 2 week in-person trainings is ideal if we can offer them to a whole region, let's say a host institution that can invite, we can invite people from the whole region, neighboring countries and so on, so that we have a larger reach. For that, we try to find funding to support our activity, but also to cover the cost for students, so that that's not a limitation. And that is what we are doing with the X3 Foundation, with thanks to a grant from Smith Futures, which is a US foundation. And for research projects itself, then we collectively look for funding to support all our Ersilia's activities and our partners' activities. Yeah, the usual suspects that fund research, not only the Gates Foundation, but also Wellcome Trust, NIH, Horizon European funding, and smaller grant projects. So I think the conditions, there are no conditions. It's having being keen on adopting these new technologies, having the ongoing research project that can benefit from them, and yeah, being open to collaborating together. We are a small organization, so we are very dedicated to helping our partners, and that's it.
Yeah, because I remember the last time you were in Cameroon, we had some students from one of them is from Chad, and he has been trying to connect. It goes on and off. I suspect the internet is messy. And also we have Basanam and Benjamin, who came from Gaundere, which is like a 6-hour drive from Boya. If they are there, they can wave the us. They are also excited to work on machine learning and do predictions and eventually do molecular docking and prediction with molecular dynamics and biological screening. Actually, the idea of our project, for those who don't know it, is that the NIGNA project, nature-inspired discovery of novel antivirals, which we are working together with Gemma and our colleagues also in Philadelphia at Vista Institute, Ntie-Kang, is that we are looking for antivirals for medicinal plants in Africa, both synthetic and synthetically modified natural compounds, and also natural compounds themselves. So in essence, we are using quite a number of approaches, and we want to build a pipeline which involves both AI, ML predictions, which are already taking into consideration potential DMPK profiling of the molecules, which will really take consideration potential toxicity in advance. Then these are the hits that we now eventually do molecular docking on them to study the interaction with the target in real time, and further take it to MD if we find some very positive binders. And then, of course, we have the possibility to screen them, biologically to find to finally verify that the predicted activities are actual. So this is already what we are doing, and we have some breakthroughs already. We have already published a couple of our results. We have sub-micromolar inhibitors already in this project, and for the AI side, we just did a paper 2 years ago, but we are already looking at what can be potentially be the outcome of this collaboration with Ersilia and work together in person. So unfortunately, our students were to go to Barcelona, did not get visas. and some of them are on the platform. It was unfortunate, but there are potentials for us to work even from it. Yeah. I would ask a couple of questions. Yes, Martin, maybe after Daniel say something about Martin's project. Looked very, very interesting from the point of view of studying the interaction between a drug and the various solvents. Maybe Daniel was planning to throw some light on this because the time was really short to give the real background for this. But maybe Daniel will say a couple of things about this aspect of the science they're doing with interacting solvents and drug molecules. Yes, Daniel, please.
Yeah, thanks. So before I say, Martin, you have something, or I go. Martin? All right. The thing is that, solvent plays an important role in practice. And the aim here was trying to understand how different solvent first would, impair the conformation of a natural product. As we know, there are several natural compounds that have different stabilities when they're exposed to different mediums. And this then has an implication in the drug design. Some other molecules, they tend even to change their polymorphism, tend even to change their nature, just because they're exposed to a different solvent. And natural product chemistry, of course, they know this well, that some molecules, you don't isolate them because of the property of the solvent. So we wanted first to understand how does the presence of solvent will change the conformation. For some that they are not shared by Martin because of short time talk. But also we wanted to understand what is the interaction, the influence of the solvent and on the interaction of a single molecule. And that we can get that information by computing the solvation free. And that solvation free energy gives more information. The synthetic chemists or to drug design people who are dedicated in exploring a range of solvents, trying to understand the behavior of a molecule. Even the conformational changes. The molecules, I don't remember the name of it, that's before years ago was used as for pregnant mothers, but it resulted a lot into a problem. It's the same molecule, but only it was changing the conformation of one of the group from R conformation to S. Once it changes, it becomes a different thing and it have a different activity. So we can explore these conformational changes once we introduce into different environments. And then we can guide this and help the other community that this molecule is behaving like this. The rest have a precaution when they want to design the molecule. Maybe in a foreign aqueous environment, the stable conformation of this molecule is like this one. But when it is exposed to this environment, it is changing its conformation. That changes should be taken into account. And it's providing very information values. Toxicity at some point. It helps more the pharmaceutical company to understand if it is going to change it from crystalline to amorphous. So that was the idea of the work.
Okay. Well, it throws more insight now because it was not too at the start, I was thinking that it was about drug delivery or access to the cells, and I was almost asking myself what's the point to investigate different solvents, knowing that the dominant solvent in our cells is water. So in essence, now it throws more light. The idea about extracting secondary metabolites from natural sources, and then we use this drug molecule as a case study. So that when you're sure about how your models work, you can now apply it generally to natural compounds. So it throws more light on this, and I think it's very clear now. Yeah, Do you guys, are there any other comments about the talks of today? Yeah. Yes,
Yeah.
Erick, please. Erick, please. Yes. Yeah. Erick, we'll get back to you, Stephen.
Okay.
Yes. Erick, you're muted. You can just unmute yourself and then we hear you.
Yeah.
Yeah.
Sorry.
Yeah.
Yes, my name is Erick. I'm a principal researcher for G&G. My role is providing risk assessment for pharmaceutical products. So I'm not really versed in the drug design process, but I study screening of the drugs and things like that. But what we have seen is that at the time we have the product in the market, when we do the risk assessment and look at things like that, it's already late. So my question to you guys, do you look at, when you do any screening or select your candidate, check for environmental impacts? Is that something that done properly or you work? I'm asking because I think there was a talk yesterday on anti-malaria drug discovery by
Samuel Gaye.
Yes. Okay, yes, he gave that talk yesterday, and I'm sure you'll be aware that antimicrobial resistance is a very, very big issue. In the EU is today revising its assessment procedures, and that is something that is going to be central in the next few years. So for this new anti-infective that we are looking at, is there any talk about how it behaves in the environment? Is that something that you can? Yeah, just
for those of you who don't know Erick, served when we were called chemical biotech too. Erick was one of the external board members of the Chemical Bioactivity Information Center. I have been very supportive of the various people looking at toxicity of secondary metabolites and all that. We're using knowledge-based approaches to predict the fate of metabolites and so on. And it was quite that. The question of the environment is something we as African scientists are not yet so preoccupied about, which is a very key issue. But we are more like, okay, the thing in the humans, because we are really overloaded by disease burden, we have issues with various diseases that are dominant in Africa. So we are more concerned about the effect of molecules in the body and in biological systems. And we forget that biological systems are also in the environment, like fishes, like algae, in marine water bodies, and so on. So by that time, we were working on building a tool, extending the Lasser's what they call Derrick at that time, for prediction of toxicity of secondary metabolite in the body. We were trying to build an eco-Derrick, or less ecological, the toxicity of compounds in the environment, and we're using chemical alerts at that time. I think it was quite an exciting project, which never went beyond the practical aspects of it. And now regulation is coming up now, these issues of microbial resistance, and which will be eventually something like what happens if you administer a medication to somebody with the malaria. Of course, the person is going to pass out urine, of course, and the urine goes to the environment, and then the metabolite of the pharmaceutical product are there in the environment. What happens?
As you mentioned, I think sorry, I have my dog here. The priority is to cure this disease. Of course, that's very, very understanding. We see a lot of people from universities or members from faculties. I think this is something that I would like to encourage the students to look at. I mean, how does the introduction of the functional group in the molecule, how does that change its environmental behavior? It's not necessarily it's something that takes a lot of time, because as you have models to screen your compound, the models as well do the screening to draw the smileys of the molecule, you put the model to tell you whether it's biodegradable, what is the potential toxic effect. So these are things that can be incorporated in today's today's training for students, because at the end, the effectiveness of these drugs, although it's important, that if you start running into antimicrobial issues in the environment, you have to balance that effectiveness, and microbial is something that is very, very important. So some drugs, the risk of taking drugs on the market, that's something that nobody wants. So that effectiveness should be balanced with the benefits of the drug. So just encouraging students or researchers to just look at this to ask yourself, how does the introduction of this molecule, how does it change the potential behavior? These are things that are learned during the process rather than at the end where the drug is on the market.
Yes, sir. So, which means, if I get it well, for every drug discovery project, it becomes interesting to see for the hits that you have, to take them further, even before thinking about taking them further to drug delivery, drug discovery, and drug delivery systems. One should really incorporate the aspect of modification of structural changes that may result from this molecule in the environment, and how will be the fate of this metabolite that will be result from it.
That's one way to look at it. I mean, I understand that at that early stage you are looking at thousands of potential, and when you start then narrow it down to target or to things that you think will go further in the developmental process, then you start looking at you start looking at that sort of issue. And just the knowledge as well, as the drug development, where I work at, our role is to provide medicines. People, that's the big
Sure, for sure. Yeah.
We cannot decouple that from the aspect of sustainability and environmental risk, which is it's not that big in tropical south.
Yeah.
In the near future, we don't need to go through the issues that we have seen in Europe or North America. We don't need to go through the same issues. We can just start asking ourselves, how do we look at it from basic chemistry, basic environmental chemistry? So that's something.
Sure. I get it very well. I think that this conversation is getting really interesting. Even though we have just 5 minutes to the next talk, it would make sense if yourself, Gemma, Daniel, and maybe Stephen, we can talk about this out of this workshop. Maybe have another meeting where we can see. For example, now we're talking a lot about AI, and we can see how much knowledge is available in this area for generation of AI models, for prediction of the fate of metabolites in the environment. And of course, we can see what are the approaches that are feasible. Daniel, if you find this of interest, this could be really a big window for opportunity of collaboration within the continent and G&D as well. So I think that Ersilia will play a very key role here, because I see Ersilia more or less bridging the gap between the North and the South. Stephen, you can find interest in this as well. Whoever wants to find interest, of course, I can share this out. Stephen, did you have something to say about this?
Yes, I had something, but I must also comment that Erick has a very good point in raising about the fate of drugs or metabolites, the environment. Obviously, when we renate or when we take drugs and then when they're excreted, they are seldom in the same state. Normally, they are being affected by the cytochrome P450, the liver. So there'll be different metabolites, but also here in the tropics. We've got UV. I mean, so this molecule will really transform into other metabolites. So I think it's worth investigating that. So yes, I'm very interested. So, but my main question was actually a comment, because we are mostly dealing with drug discovery. Yeah? The word is the word. I mean, discovery. So, but once you get some activity in the laboratory, it doesn't mean it is a drug. It has to go through preclinical trials and clinical trials. And I'm very impressed with Daniel, because he has started his own pharmaceutical company. So he'll give us insights on how he has gone through that. But my main question is to Fidele: how are we dealing, or how are we mainstreaming the issue of clinical trials? Because once we cannot get a drug onto the market unless it passes through the clinical trials.
Yeah, actually, it's a good question. Actually, right now I'm going to be attending a meeting in September where we are going to be dealing with more or less the pharmaceutical industry, the emergence of the pharmaceutical industry in Africa. Unfortunately, this meeting is taking place in Rome. It's not. I'm not saying that we are trying to install another Vatican for drug discovery in Rome. No. But what I'm just trying to say is that there's already a lot going on already, because it's a lot of molecules going in the pipeline, particularly groups based in Ghana, based in South Africa, and so on. So there is the slow emergence of the pharma industry in Africa. And of course, we know that a couple of countries have companies making generic drugs. The area of drug discovery itself has just been at the level of academia, and now there's the emergence of the industry. So it's not something that is going to be like tomorrow or after tomorrow, but it is something that is starting already. And you raised a very serious point there about UV and the tropics, which means that the models that Erick and the rest do using G&J may not necessarily apply the same in Africa, because we have a different environmental condition. We don't have winter, right? And we have different bacterial microbiomes, have different bacteria in our environment. So which means that this is a very nascent area. It's a very potentially fruitful area, because then there is either no data at all which we have to generate, or there's a data line there that we need to analyze, right? So I would say that we would like to constitute a small meeting to look at these aspects, right? The fate of metabolites and all of that could be a very promising near future project. If possible, in our next workshop, we might want to put this as a whole section on it, a whole section on the fate of secondary metabolites in the environment. That could be a very mind-blowing new venture to look at. I like to take exciting things. Yeah, that notwithstanding, we have run to the end of our session. Thank you very much for your participation, and I hope you are taking notes, and we are going to put all this together in our report, because usually we publish at the end of the meeting, we publish a report. So the report for last 2022 has just been accepted in Physical Sciences Reviews, and it's now in the press. The first year was published in Journal of Chemoinformatics, and we're hoping that this year we are also going to write a meeting report. So I will keep you guys all in touch for the meeting report. So the next talk is from Godloves Chi. Dr. Godloves Chi is a lecturer from the University of Boya, and right now he is a visiting scientist in Turkey. He has isolated some natural products, and he is also doing initiating himself into some modeling. So, Godloves, please, you are very free to share your screen and start. Are you there? Good to see you. Yeah, Godloves, I see you, but you are muted. Just unmute yourself and share your screen, please. I saw you a moment ago on video. Okay. Because the talk was originally planned for Padmika, and unfortunately he wrote in the last minute that he's not available, so his place had to be taken. So we had some some last minute slides, last minute abstract that came, and we had to replace him with Godloves. And he promised to be available to give his talk, and I think we're connected. Yeah. Yes, I hear you now. Yeah, you. Go ahead.
So I'm trying to attach my good afternoon and thank you for inviting me. Just give me some moment. I'm having difficulty dealing with a new language, so I want to attach my.
Let me give some tips. This is what you do. You're on one window, your PowerPoint is already open, right?
Yeah.
Not yet on maximum presentation, like not yet on full slide share. Then you go to the platform. Good. Now it's working. Perfect. Mm-hmm. Yeah. Because what I see now is emails I sent to you. I mean that if your PowerPoint is open on one window, you go to slide share on that, click on slide share, and when you click on slide share, you choose application window. Choose application window. And when you choose application window, second tab, you will now see the slides. You will now see your slides and you select it. That will be fine.
I'm getting some help. The computer in Turkish, so I can.
I see you. Sorry. Yeah. Definitely you'll find a way.
I'm getting some help.
Yeah. Yeah, perfect. It seems to work now.
So while we are waiting for that to be done by my colleague here, let me quickly introduce myself, even though you have already done so. So I'm Steve Godloves Group, and I'm a novice in this field. Actually, I'm from the University of Boya, I'm postdoc now at the university, at Ntie University in Turkey. So my talk will be on what is done in our lab, and all this while I've been focusing on natural products, but with no application in.
Yeah, at this point, you go to the at this point you go to the you now go back to your PowerPoint slides and click on it and then go to slide here. It will work perfectly well. I think you are almost there. If you go to the PowerPoint slide on your computer, yeah, it's back. Yes. I mean, after you had really started sharing your slides, yes, you now at this point you go now you don't touch the window where we are. You go back to the PowerPoint slides, and yes, then you now go to maximum presentation. As to say, you click on your slides where you have slide share. Perfect.
Yeah. So are we there?
Perfect. Yes, you are there now. Just click on slide share and start. You click on slide share as though you were sharing from your computer, and it's going to appear on the screen, the white screen.
Are we there now?
Almost, yes, you are almost there. So you are on the slide, you're on the PowerPoint, you're on the PowerPoint, you now do, you're on the PowerPoint, you now click on, you click on slide share. That's the button on the bottom, slide share button on the bottom. Yeah.
Can you actually see my slide? Because
yes, we can see it, but it's not light enough for cover the whole screen. So you click on it to cover the whole screen. Yes, perfect. There, just click it there and it will appear the whole screen. Because apparently it's too small for us to see properly.
The thing is, I don't even.
I think he has to put it on slide mode before
Yes.
using the window.
Okay. Okay. Are you now on slide mode?
I am on slide mode, but I'm not sure if it's been shared.
Yeah, it is. We see it, but it's not large enough for clear. Are you there? Because I see it, but it's not large enough. Yeah. Yeah.
He has to put it on slide mode before getting to the window.
Okay. This seems to be working already. It's not large to cover the entire.
Okay, I think. Let me. The problem is it's not big enough for them to see.
Yes, let it be a slide mode, then it'll be fine. Because somebody suggested that it has to be a slide mode before you start presenting. In my case, I don't have to be actually start presenting, sharing my slide, then I later on expand it. Yeah, it seems to be coming up again. Oh,
I don't love.
Yeah.
Please put it on slide mode before you share.
Yeah. I've done that.
Slide mode.
I've done that actually, but it looks like it's not.
Okay, Microsoft. Okay, anyway, go back to that. Go back to the where you were before, and go to the seventh tab where you slide through, and that will be it. Right? If you are there, just click on slideshow and then start from the beginning.
I can slideshow.
Yes. Then click on it. Click on slideshow and select from the beginning. It should work. Click on slideshow and select from the beginning. It should work, I think, because it's in Turkish. I cannot read it, but it is on mine. It is the 1, 2, 3, 4, 5, 6, 7, 8. The 8th tab. If you click on the 8th tab, I think it should be the same because yes, Turkish, I cannot read it.
8th tab.
Yes, from the top. So the first tab is file. Next one is home. Next one is insert. Next one is draw. And all that. The slideshow is to number 8. Sorry. Yeah, it was in English. It would have been easy. I think language is an issue. Yeah, because everything seems to be frozen on your computer. Not moving.
A very small suggestion to that if this is
yes.
Maybe you can just send it quickly to your email and then you can.
Yeah. Just can you send it to me? Yeah, Godloves, just send me your slides. I'll download it on my computer and share for you, and then we can go quick. I'm pretty sure that we will catch up some with the time we have left. I'll pick it up from the email. Yeah. I think it will be a lot easier to also for the next speakers in the afternoon sessions and evening. If I had your slides, that would be really great. I can actually share them for you in case you have. Right.
Have
you sent it?
Okay.
Good.
Let me get back to you. I'm actually trying to see my way through the computer. I have to okay. All right, I've gone through. Okay.
Have you sent the email?
I did. Have difficulty. I've been trying for quite a long time, about 5 to 10 minutes to upload it so that to share my screen. Actually, not been able. Yes.
Do you hear me, please? Just present now. I have shared for you, please.
Oh, okay. Thank you so much for having me, and I'm sorry for the long delays. I would like to present to you briefly what we do in our lab here in Fidele. I've just been here a couple of weeks ago, and to get it started, I was given this molecule to look at what the lab does. So in our lab, we isolate natural products and use computational chemistry approach to decide what biological activities to be carried out, and then we go ahead to carry such biological activity. Then we can determine whether the compound is good enough and include it in leads or drug discovery. So we took one of the molecules from very well-known plant called, and I can't actually move the slide. You help me?
Yes, next.
Yes, next slide.
Okay.
So we selected one of the molecules, which is from my past work, well-known from a well-known plant, Accelator. and we'll see how what goes on in our lab. So my presentation will follow this plan. We'll talk a bit about Accelator, and then application of computational chemistry, the result we got from a particular molecule I highlighted in my title, aridamine, and we'll look at discussions and what we do. So Accelator is a well-known plant. In fact, it is known as Adan in Nigeria, Prekese in Ghana, Tachokote in Cameroon. It's a very good spice we use to make the yellow soup, you know, in grass field of Cameroon. So it is used commonly in our society as cleansing for the stomach, used for weight loss, diabetes, hypertension, in other societies as childbirth laxity, childbirth. So previous studies from literature shows about 4,170 works done on the plant, Accelator. We look at pharmacological works, there are about 1,130, phytochemical work, 1,700, and chemical constituents, about 933 different works have reported chemical constituents. Now, pharmacology of this plant shows that among many other activities, cytotoxicity, antioxidant, antibacterial, anti-diabetic, anti-HIV are most commonly studied. Secondary metabolites isolated from this plant include steroids, steroidal saponins, cytopenoid saponin, flavonol, among others. This slide shows some of the compounds I isolated myself from the plant. They belong to the groups we have seen above. The most outstanding compound from this plant is aridamine, or N-acetyl glycoside. So that is the structure. And what we do in our lab is we get a molecule like what I've shown you above, with well-established structure, isolated from plant, marine meat, or endophyte, and we apply computational skills to identify what bioassays are feasible. So when we do that, this is a slide showing what we do. We get the molecules with well-established structure, isolated. We do in silico molecular docking, ADMET properties on the molecule, and then we move on to perform bioassays. Some of the common ones done in our lab include DNA protective, enzyme inhibitory, alpha-glucosidase, and others. Now, if the molecule successfully goes through this, it becomes a good compound on which other assays can be performed, maybe to clinic. Now, so what we do is actually what I'm going to show on aridamine. We first of all select proteins on which to perform the docking. So we calculate, we do 3D structures on ChemDraw. That for aridamine was developed in ChemDraw. We now apply interactions using molecular virtual doctor to develop 2D and 3D interactions using BioVia Discovery virtual program and use AutoDoc program to calculate the binding energy. So when we did that for aridamine, all doc scores are recorded as follows. You can see the picture showing 2D image for aridamine and the 3D interaction to the right. Now we compare this with standard drugs, like for this case, Doxorubicin. and you can find the result. We also did that same work for glycosidase 6-phosphate, and we obtained the result. We compared this with interactions obtained from the standard drug and glycosamine 6-phosphate, and these are the results: mole dog of 96.6, binding energy of 7.10 kilocalories per mole. Now, looking at these results, a summary which you can find on your screen, find that aridamine has comparable values to that of doxorubicin, and if not higher in the case of glycosamine 6-phosphate. We also carried out pharmacokinetic interactions using ADMET and CAiSMD software on aridamine. And if you look at the graph plotted to your right, with the void X shape in it, you find that aridamine, which is appearing some way to your right as a blue spot, shows that it is not toxic. These are results of ADMET prediction on aridamine, which we obtained from the study. So, the few slides that follow show us the different pharmaceutical pharmacokinetic properties. Bioavailability of 0.6 tells us that the molecule is very much available in the body, does not cross the blood-brain barrier. Some of these properties show that aridamine is a good molecule that could be considered for clinical studies. So this is basically what we do in our lab, and I am thankful the organizers of this seminar for giving me the opportunity to show what we do, so I could be helped by any of the persons in this community. I am also very grateful to Professor Ibrahim Demirtas for accepting me to be part of his work team. Thank you.
Right, thank you very much, Godloves. It was a big battle, but we are happy you finally made it. So, since we are already above time, I will invite Bekono, Dr. Boris Bekono, to straight away upload the slides and talk. We are sorry for encroaching into your time. Please, Boris, if you are there, please, we will take questions for Godloves in the next roundtable discussion. So you have some time to breathe after the technical challenges. Yes. Boris, are you there, please? If you are there, please share your slides and talk. He's from the University of Yaoundé one, also in Cameroon, and he's an assistant lecturer there, one of our former mentees, and we're still collaborating with the UBCDD. Please, if you are there, please upload your slides and start sharing your slides and unmute yourself. Yeah. It's the last talk before the break. If it is not available, maybe we can open the floor for conversation. I asked my co-chair to coordinate the questions that may come as a result of this talk from Godloves.
Okay. Thanks, Godloves, for let's hear some questions, comments, or suggestions from the audience. Welcome. Daniel, please unmute and welcome.
Yeah, thank you, good luck for your presentation. I saw your docking file. Usually we have some interaction called alkyl interactions. I see it in most places, but the issue is this alkyl interaction is in every protein, you know. When in lower temperature, we see it everywhere. So usually you can go to the discovery studio and actually decide to deselect that one. So you can show only the important interactions and then leave the others. And then again I saw or if you use the maestro, those kind of interaction will not show at all. Very good. So maybe your alkyl interactions, I don't know, usually the hydrogen bonds will be the most important ones. The pi alkyl is fine. But for alkyl, no, it's not any strong interaction yet. So those ones you can decide to leave them out, okay, and then maybe clear it a little bit nicer. Apart from that, okay, so basically, that was the issue I had. So basically, that is it.
Oh, thank you for that.
Thanks, Daniel. Stephen, welcome.
Okay, thanks. Yeah, thanks, Godloves, for that nice presentation. You've talked, you're studying this molecule as an anti-cancer compound, but you've not told us which cancer you're talking about, and also you've not told us which drug targets you're attacking.
Okay, thank you for your question. I'll refer you to some of our publications on this particular compound. It's a cytotoxic compound which we tested already against leukemia cancers. So there is a publication that shows this molecule is very active against leukemia cancer cell lines. And in some cases better than doxorubicin, the standard drug we use in this presentation.
What about the mode of action? How does it work?
It works through ferrotoxicity. I think for clarity on what we do, I'll send to you our publication on this ferrotoxic molecule, and you can read about that detail.
Okay, there are many questions here on the chat. Maybe before Fidele you talk, can I mention this question? Then you will leave.
Yeah, of course.
Perfect. Let me read some of these questions for you. Thank you for your presentation. Please, how do you determine the RD50 using Swiss ADMET?
The RD50?
Yes, using Swiss ADMET.
Okay, I must say I would not be able to answer this question because I'm still new to this software. I've just been a couple of weeks introduced into them. I would not be able to answer that. Maybe Professor Fidele could help us on this.
I was just about to answer on the chat. Actually, what Godloves has done to using two web servers, or two well-known web servers, which I would encourage for everybody who is interested in GAPK or anything like ADMET. You use Swiss ADMET is available, and also PKCSM. And we also routinely use these two software, these two web servers. So the prediction of NFT values is done PKCSM and not with Swiss ADMET. So if you get a smile from your compound and you go and you upload the smile on PKCSM, you have the possibility to either run just the initial baseline tox profiling or baseline ADMET profiling. And then also you also have that will just calculate parameters like Lipinski, Weber violations, and so on. Then if you want to do a complete study of your molecule, you can actually click on ADMET. Then it gives you everything, including prediction for inhibition of enzymes. It gives you prediction for toxicity in human oral toxicity. It also gives you the possibility of doing toxicity for extrahymenal bioinformatics.
Sorry, Fidele.
Yes.
Can I ask you related
yes,
to thanks. Please go ahead.
So it also does toxicity for against prediction against tetrahymenal bioinformatics, a fish species typically used for modeling toxicity environment. Also, fat minnow is another fish species. So it gives you a couple of other additional data that you don't have from Swiss ADMET. So I think it's PKCSM that does this.
Thanks. There is another foreign question from Rob. I think why? Maybe I can rephrase. Why did you specifically selected one compound? You know, synergistic effect is crucial when evaluating a brand for drug discovery.
That's a good question. Thank you for that question. If you followed up yesterday, I had the same impression in mind, and I asked that, can we verify that? And I think that is one of the outcomes of this seminar that we carry along. I did that because this is what I can do right now, what the software allow me to do. Maybe my knowledge of using the software is still shallow, and I don't know how to do that for more than a single molecule. But to follow conventional approach, we must work with one molecule able to determine every pharmacological aspect of molecule for moving things of combination of.
Okay. There's another question. I think this has been answered, but I can mention it. What were your target and bacteria cancer cells? I think you already answered it. The other question is, what was the importance of using outdoor Puvina and MDV? And I have an additional small question on it. What are the units of Murdoch score? Can you see for outdoor Puvina is kilocalorie per mole? What about the Murdoch score? Those are the questions. Welcome.
Okay, let me start with the last one. I think Murdoch has no unit.
Really?
Best of my knowledge.
No. Better check it. I think there should be a unit. Check it. There should be a unit.
Right?
Yeah.
And the other question was, what was the importance of using photodopsina? I think it's important to do this on different softwares, find which one is most appropriate. And I would say these are the softwares we have available or the softwares I have been exposed to as of now. So that is why I made use of them. But in the future, when I get into this field better, I'm sure I'll be able to compare different software better than the other. For now, I have just an introductory knowledge with the using of this software. All right, thank you.
Daniel, you are raising your hand. So Daniel start and then Fidele will.
Okay. Just a comment on the move dog about the unit. I use the Go software, and the Go software gives what we call scores, which is a measure of binding affinity. So it doesn't give unit.
I see.
Yeah, so the Go doesn't give unit. So sometimes when I present this argument comes up, but I'm personally aware that some software set up the Go doesn't give unit, but the Autodoc gives. So so you should not be so strict. Some give, some don't give. But I don't know really for sure for the moment. But just to draw attention that some give, some it's just a score. Thank you.
Thanks. Fidele?
Yeah, thank you very much. I think it was I just wanted to say something brief before we go on break. When you present the numbers like LD50 values and so on, it might be nice to also put a footnote to state which cutoff value is interesting for a compound that is not toxic. If you say, for example, LD50 is this number, then you specify that above this cutoff value, compound is considered to be toxic. If you say for all the, and this applies the same for all the pharmacokinetic parameters you have. And of course, it would have been nice also to show the CYP inhibition, because typically this is a nice thing to see about any compound, beyond showing the volume of distribution and the half-life, the half-lives. So that's basically what I wanted to say about that. And for those who came at the beginning, at the end, know that the Godloves is trained as a nitro-product chemist, and I really encourage his boldness to confront a new area of research. And of course, he's coming back to Boya, and we have at the Center for Drug Discovery a lot of tools that can be very useful for his further training from Richland. So thank you very much for participating. Thank you very much, Daniel, for co-chairing this session. We are break time already, and we are coming back in 2 hours time. We are coming back. My timetable tells me that we are coming back from break at 12 o'clock. No, at 2 o'clock. Sorry, and we will be starting with a keynote lecture. I encourage you very much to be part of it. He's a professor from Brazil, and with a lot of experience in natural product research, and they will be sharing their experience. And I think they are going to inspire us in who are also living on the continent. Don't forget that biodiversity in South America is very common, similar to that of West African coast, because of we were one big continent before the Atlantic came between us. So encourage all you guys to have a good time, good break time, and we look forward to meeting you at 2 p.m.
Thank you.
So bye.